Okay, good morning, everyone. My name is Felix Kugler, and I have the pleasure to introduce Stéphanie Manel today. Stéphanie is a fellow in our research unit, Biodiversity and Conservation Biology. She had 3 stays here at WSL, and it seemed that whenever the virus was increasing, Stéphanie was coming, not the other way around. So Stéphanie did her PhD at the University of Lyon and then became ingénieure d'études at the University of Pau. Later on, she moved to Grenoble, to the Institute LECA, where she was maître de conférence. And it was more or less at that time when Stéphanie became Mrs. Landscape Genetics due to her work and the seminal review that she published in 2003. And at the same time, she also moved further from landscape genetics that looks at barriers to gene flow. She moved to landscape genomics, in which adaptation to environment has become the key topic at that time, at least. Later on, Stéphanie moved to the University of Marseille, and in 2014, she moved to the place where she's staying now. She became professor at the École pratique des hautes études in Montpellier. And along this route, she has moved from alpine plants to marine organisms. I guess these are more palatable than chewing on herbal species. So, Stéphanie, I guess your main topic today will come from the marine side, and I look forward to hearing your presentation.
Thank you, Felix. And not only marine side, but let's see what happened. First, hello everyone, and thank you to Felix and Rolf to let me the opportunity for this fellow in the WSL. And today I'm very happy to be present and to present this talk. I'm going to illustrate how landscape genomics can help to study the genetic response of species to climate change. Oh, sorry, I forget to share the screen. Okay. Okay, this is so to predict, yes, and climate change is a stress for biodiversity, and everyone knows that at this point. And one manifestation of this stress is shift in the geographical range of species over time, and this is what is shown on this figure. Indeed, you have the solid line represent the species distribution of one species in relation with latitude, and the dashed line represents the species response to future climate change. And you can see that climate change causes range contraction and population extirpation at low latitude, and indeed expansion beyond the leading edge that is called edge. And so landscape genomics, that is the association between climate and genome, can help to identify adaptive genetic variation and then to identify local adaptation. And genetic variants confer adaptation to environmental extreme, and we would expect their frequencies to change along the corresponding environmental gradient. And if we can capture this signal in current genomic climate association, we can then use it to predict genomic vulnerability under scenario of change across the same environmental gradient. And here you can see that I'm move from genetic to genomics, and it's just because we will use a high number of genetic markers to address the question. And so, depending on species, the genetic response is not the same. For example, long generation time species may have difficulty keeping up with the rapid climate change, and they are prone to local extinction in their current main elevation range. And this is the case, for example, for Swiss stone pines, and that is published in the nice paper of Benjamin Dauphin here et al. But we can ask what happened for other species, for example, for marine fish with a short generation time, or for bats with a medium generation time. And this is what I'm going to illustrate here today. I'm going to illustrate from two examples how landscape genomics can help to detect the species, the genetic response to climate change. And first, I'm going to present this example that is published in journal Animal of Ecology, and that investigate the genomic pattern of two marine fish. And this is a part of the PhD of Émilie Boulanger. And indeed, this is just the first part of the story by looking how to correlate genomic variation to the climate. And for that, I'm going to illustrate the association of genomic variation with climate from two exploited marine fish, the white sea bram and the striped red mullet. And both species inhabit rocky and sandy coastal shores across the Mediterranean Sea and Eastern Atlantic Ocean. And they spend their first months of life as planktonic larvae that can disperse with oceanic current. And we have conducted a large sampling across the Mediterranean Sea, from the western to the eastern. And in collaboration with fishermen, we have collected tissue in about 50 sites, as indicated here by dots, and that conduct to have about 400 individuals for the red mullet and about 300 individuals for the sea bram. And then we applied a genotyping by sequencing approach to each individual to derive a high number of genetic markers that are called here single nucleotide polymorphism, and that correspond, as you can see here, to the variation of one nucleotide of each genome sequence. And then we separate the SNP that are located within gene or just close to a gene, and they are called adaptive SNP, from the neutral one that are in non-coding region. And then in the next of my talk, I'm going to focus on the adaptive SNPs, and I'm going to illustrate some results about the influence of climate on those adaptive SNP, since they might indicate local adaptation. And so the next step is to associate this genetic variation quantified from the SNP to predictors. Then to quantify genetic variation, we estimated pairwise genetic distance between individuals. And then for the predictor, we consider three categories of predictor: the environment with sea surface temperature, salinity, and chlorophyll. We also consider larval dispersal, that is measured by asymmetric eigenvector map, very technical. I will not enter in the detail. And we also consider space. And we applied a distance-based redundancy analysis, that is a multivariate analysis that combine multiple linear regression between the genetic variation and the predictor, and the principal component analysis. And then I'm going to show you the result for the mulus. That is indeed the result of the principal component analysis. And so this figure represents each dot is represent the genotype variation of an individual in the plan 1 and 2 of the axis, the PCA. And indeed, globally, the DBRDA, the analysis explained 8% of the genomic variation with the three category of predictor. And what you can see here is that we have here one group of individual that is associated with a variable of dispersal and space, and negatively with the summer temperature. And this group of individual, indeed indicated is by the yellow arrow, correspond for the individual that are located here in Gibraltar, and that is the more cold part of the Mediterranean, since the Mediterranean Sea is characterized by a climatic gradient and increase of temperature from the western part to the eastern part. And then a second interesting result is here, this group of individuals that show that genomic variation here is associated to chlorophyll and negatively with the winter temperature. And this group of individuals is here more specific of the Adriatic part of the Mediterranean Sea. But indeed, you can see here that environmental variation is confounded with spatial and dispersal variation. And to separate the different effects, we apply the partial DBRDA, indeed, with a law to separate the effect of the different variables. And the results are represented on this figure. Indeed, the surface of the different section of the diagram is proportional to each exploratory power of each type of variable. And what you can see is that indeed environment explains 80% of the genomic variation. So, and the next step to go further in the landscape genomics is to look how those climate-associated SNPs are located within gene that code for a protein. And in this example, for the mollusc, we found 8 candidate SNPs within a gene that code for a protein. And indeed, here on this figure, I represent an example of one of these SNPs that is associated to a specific protein, the tupulin specific chaperone. And you can see here that the black genotype indeed is present only below 70 degrees. And so that means that the population with this genotype will be in the western side of the Mediterranean, that is more cold. And the red genotype is present everywhere, but more frequent in the warmer part of the Mediterranean Sea. And so here we can say that we reveal indeed population adapted to the cold and population adapted to the hot temperature. So in this marine example, I illustrate how genomic variation can be associated with climate and reveal local adaptation. And I will present now in a second example how we can use the genetic response to make some prediction in relation with climate change. And here I'm going to develop this example on bats, on the Myotis escalari. This is a work that has been done in collaboration with Orly Rasbourg. And here we have looked for the species response to climate change, accounting for adaptive genetic variability and species dispersal ability in the landscape. And we follow this methodology indeed. First, indeed, we have developed ecological niche model on occurrence data only. And we then did future projection, and we are able to estimate range loss prediction for the species. And then to account for the evolutionary response, we work from on genotypes this time and on genetic marker. And we first look for SNP associated with climatic condition. And then we were able to separate hot, dry population from cold, wet population. And then we applied species distribution model for each type of population. And we were then able to predict range loss prediction, accounting for evolutionary response. And we were also able to look using genetic data, the population structure and the movement of the species in the landscape. Indeed, in a more practical way, we analyzed about 200 bats in 18 colonies and derived about 18,000 SNPs. And here you can see one part of the result that we get indeed on the left with this map. The map represents the species response, considering only occurrence data, and in first the present, and with blue color indicated low species occurrence and green color medium and red color high species occurrence. And below you have the future projection under a scenario of climate change, and you can see that the population moved to the northern area in the Europe, and indeed disappear from Spain. Now, if we look for and if we compare the response accounting for the genetic data, here you have in red the hot dry population, in blue the cold wet population, and in yellow the both type of population. Indeed, you can see that and first always here this is the under present condition and below the future condition. You can see that most of the blue population adapted to the cold disappear, and indeed, and they were replaced by the cold dry population. And indeed, if we compare the habitat lost between species response only and when accounting for the genetic response, we show that considering genetic intraspecific diversity reduced habitat loss in comparison to classical niche models. But indeed, to fully address the question, indeed, we also look if the how population were able to move in the landscape. And this is done using some what is called landscape resistance model. And we show that the white circle that are the hot, dry population were able to reach the gray circle that represent cold, wet population. So that means that it's possible that genotype adapted to hot condition move to the other place. So now I have finished with the example, and just I want to ask the question, is landscape genomics ready to make such projection to future climate change? And indeed, this was the main reason of my stay here at VSL. It was to have brainstorming or where we are in landscape genomics. And indeed, we focus on two major points here at VSL. First, the first point was to discuss about how to make landscape genomics more predictive. And the second point was to develop new approach to make population genetics. So I'm going briefly to explain. For the first point, indeed, we are writing a review and perspective paper that will discuss the main point that where we can work to make landscape genetics more predictive. That is improving the estimation of genomic variability, improving the sampling design, improving the spatial and temporal resolution of environmental data, and adding more explanatory variables like biotic variable, and improving the model. And so we have this discussion with a small group of people, and sorry, we still have not finished. And so it will be probably the topic for next talk. And for the second point, it's indeed the idea is to investigate the potential of environmental DNA for population genetic analysis, and I hope in the future population genomics. And indeed, for example, in marine ecosystem, it's very difficult to sample genetic tissue, and for example, you cannot go in reserve, and so this is why we want to look if it's possible to use environmental DNA for such study. And this is a collaboration with Loïc Pellissier and the Launch Science Unit. And here we have an INR that is just funded, Shift DNA, so in collaboration with the VSL and the Museum of Geneva. And we also submit a biodiversity project to investigate the use of eDNA at intraspecific level. And so, thank you. This is the end of my talk.